we will present paper 140 called Allogeneic BMP6 gene-modified bone marrow mesenchymal stem cells induce vertebral fracture repair in a porcine model, a pilot study. This is presented by Dr. Bay. Thank you very much. I'll answer your question.
Okay. My partner's here, so you can answer it. Okay, so this is from Cedars-Sinai. We have a regenerative skeletal laboratory where we're really looking at stem cell therapies for regenerative therapies for the skeletal spine as well as long bones. These are my partners. And right now we're looking at vertebral compression fractures. One of the targets that we have is to develop a stem cell therapy for the treatment of vertebral compression fractures. Now, just as a background, mesenchymal stem cells, they are pluripotent in the sense that they can develop into cartilage, bone, muscle, skin, and other tissues. And a lot of times when we think about stem cells, there's a lot of hype with them. And just to tell you, in our experiments with stem cells, we haven't really been successful without trying to modify them in some way or give them some type of kick to make them work. So if you take a massive bone loss, let's say a femur fracture, or here we've modeled into a radius fracture, and you use stem cells alone, we found that we really don't get very much bone regeneration, if any. So we tried to use gene therapy in making sure that these stem cells kind of progress down the osteoblastic lineage. So we've engineered the stem cells typically by modifying it or inserting a gene, and then we get very, very good bone regeneration. This is a little bit of a background. This is a rat vertebral bone defect, and we were trying to compare the differences between using BMP2 as a gene or BMP6. You can see that we can recreate the vertebral body defect with mesenchymal stem cells that are modified with BMP6. We could do this with BMP2, but not as well. But stem cells alone just really don't have that capability. So one of our hypotheses is if we have a fracture in the vertebral body, can we use stem cell therapy and maybe even modify the stem cells to make sure that we do get the type of healing that's required? So our experimental design, we used pigs, mini pigs. The stem cells were bone marrow derived, and it's basically a plastic adhesion type of assay. We genetically engineered them with plasmids. We wanted to stay away from viruses. So for clinical use, we really think that viruses aren't going to make it to the clinic. There's an immunogenic reaction. There have been some problems with very severe illnesses due to the virus, the reaction to the virus in human clinical trials. Plasmids, it's just DNA. They stick around for maybe about 2 weeks, and they're kind of gone. Maybe peak plasmid expression is really within 3 to 4 days, and then they disappear. So we like this idea. We used electroporation. Here's our critical size defect. It's basically a bone void in a vertebral body in a pig. Through a lateral approach, the bone void is 4 by 15 millimeters. And we used a fibrin gel as our carrier. So these are our groups: BMP2-engineered MSCs at the concentration of 4 times 10 to the 6, BMP6-engineered MSCs. And these are all done with plasmids. And then fibrin gel alone, which was just the carrier. And if you look at our results 6 months post-surgery, you can see the critical size defect above, where you can still see the defect. And the lower slide, or the lower picture, you can see nice healing of that defect with our genetically modified MSCs. This is just a MicroCT by bone volume. Again, fibrin gel alone, MSCs modified by BMP2, and then MSCs modified by BMP6. And BMP6 performed the best in our pig study as well as in our rat study. If you look at histological analysis, again, we really see good recreation of the trabeculae from our bony defect. We really didn't see any inflammation or no bony overgrowth, and that's really what we were looking for. So in conclusion, allogeneic BMP6 gene-modified MSCs induced bone regeneration in a pig vertebral defect model. Bone formation was enhanced compared to the vertebrate treated with the BMP2 modified MSCs or fibrin gel alone. And we saw no signs of neurologic side effects, bone growth in the spinal canal, or immunogenicity. Thank you very much.